guys, welcome back to another ASMR study session. In this video, we're going to try and do units 3A, 3B, 3C, 4, and maybe 5 if I have energy and if I have time. But we're going to start by reviewing just a few miscellaneous items like claim, evidence, reasoning. Just remember for your claim, evidence, and reasoning. Claim. Does the claim answer the question? Is the claim a complete sentence more than a yes or no answer? For evidence, is each piece of evidence relevant to the claim? Is evidence listed specific and not vague or generalization? In reasoning, is there justification for how the evidence supports the claim? Also, an example would be like a claim: double stuffed Oreos have double stuffing. It's a claim. Evidence: in the graph, the normal Oreo has an average stuffing mass of 2.83 grams, and the double stuff average has a mass of 6.74 grams. The standard error of the mean is 0.12 for both Oreos, meaning that in all possibilities, the double stuffed Oreos are always double the weight of the normal. And the reasoning would be: matter has mass. Double stuffed Oreos have double stuffing. If they have double stuffing, then they have double mass compared to the standard, and they are double stuffed. So reasoning is pretty much like dummy so far. What is a positive and negative control? A positive control, you're expecting a result. It's a control where you expect a result to happen. A negative control is where you're expecting a result to not happen. And if positive has a negative reaction or negative has a positive reaction, then the control will show that there's going to be an error in your experiment. And properties for water. Why is water a universal / excellent solvent? Because water is a polar molecule due to polar covalent bonds between the oxygen and the hydrogen or the electrons. Oxygen is more electronegative, which results in a negative charge on the oxygen and a positive charge on the hydrogen. And then the oppositely charged poles of water form hydrogen bonds with the charged solutes, like salt. And the adhesion of water to charged solutes results in the solutes being dissolved to the hydrogen bonds, charged solutes. Why is water a good way to moderate temperature within living organisms and in an organism's environment? Because water has a high specific heat. It takes a lot of energy to heat water because the hydrogen bonds between the water molecules must be broken. And heat is absorbed when the hydrogen bonds are broken. And this allows water to serve as a coolant within the cell, organism, and planet. Like sweat is an excellent coolant because water acts as the heat sinks in the environment. And describe to me the process of capillary action in the movement of water from the roots to the leaves of the plants, and after how it gets out of the leaves of the plants. It uses cohesion and adhesion and capillary action to get up to the roots, up in the roots in the plant. Adhesion sticking to the other charged molecules / the sides of the tubes, and cohesion sticking to itself, pulling each water molecule up, building on top of each other, using capillary action, going against gravity, going up to the leaves, and then using transpiration or / circulation of water through the leaves, and cohesion it pulls the water molecules out onto the leaves, up through the roots of the stems to the leaves. Now on to Unit 3A, Enzymes and Energy. What are enzymes made out of? Proteins. What do enzymes do? Help speed up metabolic reactions / chemical reactions. What happens in an enzyme? Like, how does it work? The enzyme has a substrate nearby, and it takes on the substrate from the nearby area and catalyzes the product / breaks it, and then releases the enzyme product, which can either be broken from the substrate or the synthesis of the substrate. What are two environmental conditions that can affect the activity of the enzyme? Temperature and pH. How does temperature affect an enzyme? Because the increase in velocity and kinetic energy that follows temperature increases. With faster velocities, there will be less time between collisions. This results in more molecules reaching the activation energy, which increases the rate of reactions. So the higher the temperature, the more kinetic energy and velocities between collisions, more molecules reaching the activation energy. Speaking of activation energy, how does an enzyme speed up a reaction? It lowers the activation energy needed to do the reaction. Are enzymes used up or changed during a chemical reaction? No, they're not. What is the region where the reactant binds to the enzyme? The active site. Fill in the blank. Enzymes are biological blank that speed up chemical reactions. Catalysts. They are biological catalysts. Enzymes are the perfect shape to fit their blank substrate. They fit like a blank. It fits their specific substrate / fit like a lock and key, and they can be used over and over again. High heat or pH changes change the shape of the enzyme. If the enzyme changes shape, is it still able to function? No, it is not. Enzymes are protein molecules that are made up of long chains of blank amino acids. And when the enzyme changes shape, what happens to it? Like, what is this process called? We talked about it last time. When it gets deformed, it gets denatured. I'm going to go through and list different questions, and you're going to tell me whether it's catabolic or anabolic reactions. Which reactions release energy? Catabolic. Which reactions consume energy? Anabolic. Which reactions build up larger molecules? Anabolic. Which reactions reactions break down molecules. Catabolic, which reactions are considered uphill. Anabolic, because they use energy. Which type of reaction is photosynthesis? Anabolic, which type of reaction is cellular respiration? Catabolic, because cellular respiration yields energy, but photosynthesis uses energy to produce sugar and oxygen. In living systems, which reactions require enzymes to catalyze them? Both. What is free energy? And what is its symbol? A portion of the system's energy that can perform work when temperature and pressure are uniform throughout the system. The symbol is known as G. Once we know the value of G for a reaction, we can use it to predict whether it will be spontaneous. For an exergonic reaction, outer is G negative or positive. G would be negative because there's a net release of free energy because of the chemical mixture. What is the first law of thermodynamics? Energy can be transferred and transformed, but not created or destroyed. What is the difference between kinetic energy and potential energy? Kinetic energy is the relative motion of objects, and potential energy is the energy that the matter can possess due to location of the structure. What is a spontaneous process? This is a process that occurs without an overall input of energy. It is energetically favorable, and there's an increase in entropy, which is randomness. Is cellular respiration endergonic or exergonic reaction? Cellular respiration is an exergonic reaction, and it has a negative G symbol or free energy symbol. If energy is released, G must be negative. Is photosynthesis endergonic or exergonic? Photosynthesis is endergonic. And what is the difference between endergonic and exergonic? Exergonic reactions are also called spontaneous reactions because they can occur without the addition of energy. But photosynthesis needs energy. So there's going to be an energy source that drives it, which would be the sun. It's a chemical reaction. What is energy coupling? It is when energy released from an exergonic reaction is used to drive an endergonic reaction. So like if energy released from cellular respiration was used to drive photosynthesis. And what kind of work does a cell do? Chemical work, like pushing of endergonic reactions that won't occur spontaneously, like the synthesis of monomers to polymers. Transport work, pumping against concentration gradient like the sodium-potassium pump. Active transport, and mechanical work like the movement of muscles, movement of chromosomes during cellular reproduction, like meiosis or mitosis, which we will try to talk about. In many cellular reactions, a phosphate group is transferred from ATP to some other molecule in order to make the second molecule less stable. What term is used to describe the second molecule that receives phosphorus from ATP? Which would that be? I don't know. Phosphorylated intermediate. If you could not regenerate ATP by phosphorylating ATP, how much ATP would you need to consume each day? It says if ATP cannot regenerate through phosphorylation of ATP, humans would use their body weight in ATP each day. Maybe just an FRQ to have in the back of your mind. What is a catalyst? A chemical agent that speeds up a reaction without being consumed by this reaction. It doesn't get consumed. What is activation energy? The amount of energy that reactants need to absorb before the reaction starts, aka free energy of activation. What effect does an enzyme have on activation energy? It catalyzes a reaction by lowering the activation energy barrier. And is G positive or negative here? G would be negative. And how is G affected by the enzyme or the free energy? G is not affected by the enzyme. It cannot change the G, the free energy for a reaction, and it cannot make an interconic reaction exergonic. Exergonic being spontaneous, it can. Reaction that need pushing are going to always need pushing. What is the substrate? It is the reactant at which the enzyme works. What is it meant by induced fit? What is an induced fit? It's when the enzyme changes the shape of the active site so the substrate bonds more snugly to the enzyme. What are a few ways that enzymes lower the activation energy? I'm looking for if there's two or more reactants, the active site provides a template on which substrates can come together and have a reaction. There may be a microenvironment that is more conducted to a reaction. Amino acids may directly participate in the chemical reaction. And when an enzyme clutches the substrate, it can break bonds that must be broken during the reaction. How does protein structure / amino acid structure involved in enzyme specificity? Because enzymes are proteins have a unique three-dimensional shapes from primary, secondary, tertiary, and quaternary foldings in the amino acid sequence, specifically like a lock and key form to the substrate function. Form and function. How does the initial concentration of a substrate affect the rate of enzyme action? The more substrate available, the more access there is to the enzyme to the active site. So there'll be more reactions. What is a human enzyme? This is like a good example to have in the back of your head that functions well in pH 2, and where is it found? Pepsin, like Pepsi, but with an N. It is a human enzyme found in the stomach. It's a digestive enzyme. What is the difference between cofactors and coenzymes? A cofactor is a non-protein molecule that is required for the proper functioning of that enzyme. They can be temporarily or permanently bound to the enzyme and be an inorganic like zinc, copper, and iron. A coenzyme is an organic molecule as a cofactor like vitamins. Is an organic molecule. So cofactor is a non-protein, non-organic molecule that is required for the proper functioning. A coenzyme is an organic molecule / protein like vitamins. What is the difference between competitive inhibitors and non-competitive inhibitors? Competitive inhibitors are substances that reduce activity of the enzyme by entering the activation site. They're competitive because they're fighting for the activation site, and non-competitive inhibitors reduce activation by binding far away from the active site and still change the shape, making the enzyme least effective. So as you see here, this is competitive because it's in the active site right there. It's in the active site, but non-competitive is here because it binds away from the active site but still changes the shape of the active site. Describe how pH and temperature may affect an enzyme, not bonds and stuff, but specific enzymes, how pH and temperature might affect those specific enzymes. Those specific enzymes have specific pH and temperature ranges at which they can best function. Many toxins and poisons cause irreversible enzyme inhibition. What is an example of why toxins and poisons can be deadly? Like sarin nerve gas, it binds covalently to the R group of an amino acid serine, which control nervous system processes, making muscles move uncontrollably. So it binds to the R group of amino acids, which control nervous systems. Sarin nerve gas is an example of enzyme inhibition. What is allosteric regulation? This is the binding of a regulatory molecule to a protein at one site that affects the function of the protein at a different site. How is allosteric regulation like non-competitive inhibition? Because non-competitive inhibition binds at another site affecting another, but allosteric regulation can have a positive outcome instead of a negative one. So allosteric regulation is not inhibition, it's just the binding of a regulatory molecule, like a protein, that can affect the function, good or bad. What is the difference between an allosteric activator and an allosteric inhibitor? Activators stabilize the shape and function at the active sites, and inhibitors stabilizes the inactive form of that enzyme. And either can turn the enzymes on or off. starting with, okay, tell me the cellular respiration. What are the 4 reactions that give you, and what does this give you? Cellular respiration is used to get energy / ATP, and the 4 processes are glycolysis, the link reaction / acetyl-CoA reaction, they're the same thing, the Krebs cycle, and the ETC or electron transport chain. Starting with glycolysis, what goes into glycolysis? Glucose, 6C, 6 carbons, 2 ATP, and 2 NAD+, and 4 ADP. It starts out with the energy investment phase where you put in the ATP and the glucose, and it breaks the glucose into what? Into 2 3-carbon molecules, and it takes phosphate from ATP. And what does reduce mean? Oxidate, reduce. Oil rig. Oxidative is losing, reducing is gaining. Oil rig. There's also no CO2 waste in glycolysis. After it gets the phosphate from ATP and it reduces NAD+ to NADH, then you get the 3-carbon molecules with 2 phosphorus, transforming 2 ATPs into 2 ATPs, leaving you with 2 3-carbon molecules of 2 pyruvic acids. And the middle, the energy investment phase, starting with glucose and putting in 2 ATP, is an endergonic reaction. Because you need energy, it's not a spontaneous thing to happen. Exergonic, because spontaneous is the adding phosphate from ATP, reducing NAD+, to NADH, to an exothermic reaction of taking the phosphorus and phosphorylating ATP to ATP. And what is oxidative phosphorylation? Because that's what's happening. That's what's happening right here. Actually, right here, it is using substrate level phosphorylation, and that uses enzymes. Okay. And this gives you how much ATP? 4. And where does this happen? The cytoplasm. That is correct. It is an anaerobic reaction because it doesn't use oxygen. Moved an electron when breaking glucose and gives it to NAD. This is what it looks like. Starting with glucose, breaking it down. It takes an electron and adds it on to NAD+, making it NADH. Getting phosphorus from the 2 ATP in the beginning, putting it there. Breaking it down, making the ATP, giving you 2 pyruvic acids, which go into the acetyl-CoA link reaction, which takes what? NAD+, 2 NAD+, and 2 pyruvic acids, which is 2 3-carbon molecules. Where does this happen? The mitochondrial matrix, which is the inside of the mitochondria. To does this produce any ATP? The acetyl-CoA link reaction? No, it makes 0 ATP. It just breaks the pyruvate into a coenzyme, a CoA, and 1 CO2. So there's 2 CO2s that it makes. And then using the pyruvate, it takes oxidation to take it into the CO2 and the CoA coenzyme, and reduction of NAD plus to NADH. So it gives you 2 acetyl-CoAs, 2 CO2s, and 2 NADH. Where does this NAD plus come from? Yeah, it's just kind of like floating around, almost like it's not put in from anywhere. It's kind of like if cellular respiration was an e-bark, NAD plus and FADH would be the shuttles taking people, the electrons from each place, like a taxi almost. They're like just carrying electrons throughout the process. What is the next reaction that takes the 2 acetyl-CoA and a few other things? This would be the Krebs cycle. This takes 2 acetyl-CoA and it takes 2 ADP, 6 NAD plus, and 2 FADs. Where does this happen? This happens in the matrix, the mitochondrial matrix. Is this an aerobic reaction? Yes, because it uses oxygen. Oxidation here. Same with link reaction. It's an aerobic reaction. Starting off with the 2-carbon acetyl-CoA. What happens next? Or just think about what happens. It adds a 4-carbon oxalate acceptor, which turns it into a 6C citrate. And then 2 of these carbons get oxidized and taken off. So it goes back to the 4C acceptor that kind of transports it to the oxidation, where it turns it into 2 CO2s. And then at substrate level phosphorylation, it turns the 2 ATPs into ATPs. There's a reduction of FAD to FADH2. And Newton's third law of coupled reactions. Newton's third law, that's what it is. When something is oxidized, something's reduced. So because there was 2 things oxidized into 2 CO2s, the 2 carbons, there was 2 oxygens added. Oil rig oxidation is losing. The 2 carbons were lost. Oxygen was added to 2 CO2s, so now there's 2 reductions. So NAD plus now to NADH. So now we have 6 NADH because 3 NAD pluses. There was regeneration there, so just take a look. Honestly, just pray that this is not on the exam. If you want to just look at this, just remember it starts with the 2 acetyl-CoAs. You get 6 NADHs at the end. You get 6 FADH, or 2 FADH times everything by 2 because there's 2 acetyl-CoAs to start it with. And then you would get net total 2 ATPs because 2 ATPs are put in to make 2 ATPs. And this leads to the last reaction, which has the most energy of how much energy does. Okay, what is the last reaction? The electron transport chain / the ETC. How much ATP does this produce? 32 ATP. And this happens where? This happens in the inner membrane of the autocristae, which is like the matrix membrane. And it goes from the inside the matrix to the outside. Still inside the mitochondria, but it's not the inside there. It's right in this little lighter part. How does the electron transport chain work? Using a concentration gradient, there's potential energy, like we talked about, the building up of ions to one side, using a pump. Because the proteins are embedded in the membrane, and how many NADHs and FADH2s are put into this? Are put into this, which if you add up all the past NAD pluses we've used that turned into NADHs, it would be 10, and FADHs, the past 2 FADHs that we got, goes to this. So they carry the electrons here, and using those electrons, it drives the H ions, the hydrogen ions, from low to high, not high to low, which is the normal concentration gradient. As the electrons go from each pump to pump, pushing the hydrogen atoms up, and then once the electrons get off, half an oxygen is used as the final. Oxygen is the final electron acceptor here. And then it accepts the 2 electrons at the end to make a water molecule. So eventually, 2 hydrogens, and then half an oxygen molecule. So, because half an O2 would be an O, H2O, oxygen final electron acceptor. And this is a redox reaction because oxidation happens with the NADHs and FADHs, leaving you with 10 NAD pluses and 2 FAD pluses to put back into the system. And then reduction happens with the addition of 2 hydrogen to the oxygen to make the water as the waste. So this has an H2O as waste. There's no set amount for that. And what is chemiosmosis here? It is when these hydrogen molecules just kind of fizzle through the membrane with no pumps, going through the high to low concentration gradient. What is the proton motive force? It is when there's the concentration gradient of going high to low, and we can definitely talk more about chemiosmosis. I'll describe it better later. What is ATP synthase? And how is it used? this is where oxidative phosphorylation happens. It adds a phosphorus to the ATP to make it ATP. So 32 ATPs are put in to give you 32 ATPs. And oxidative phosphorylation can only happen in the presence of oxygen. So if there's no oxygen, then what happens? It stops making ATP. Describe to me what cyanide poisoning or rat poisoning might do to this chain. It bonds to the O2, so the electrons cannot be finally accepted. So when they're going from pump to pump, and then they would normally be finally accepted to form a water molecule and leave, there's nothing to accept it, so the electrons can't keep going, and it just stops. It backs up the whole system, halting ATP production. And oxidative phosphorylation. Chemiosmosis is the H plus protons flowing down the concentration gradient from the inner membrane back to the matrix. H plus ions flow from high concentration to low concentration through the ATP synthase. This powers the synthesis of ATP. Oxidative phosphorylation happens because ATP is phosphorylated to ATP, while O2 serves as the final electron acceptor. So that's why it's oxidative phosphorylation, because oxygen is the final electron acceptor. And NADH and FADH, so what do they deliver to the ETC? Electrons. They're electron carriers is what they are. Okay. Tell me where glycolysis happens. Cytoplasm, link reaction, mitochondrial matrix, the Krebs cycle, the matrix, the ETC, the inner membrane of the autocrista going from the matrix to the cytoplasmic space, intermembrane space of the mitochondria. And which reactions are aerobic and which ones are anaerobic. Glycolysis is anaerobic. Everyone else is aerobic. Which ones provide ATP and how much? Glycolysis provides 2 ATP and 2 total, 4 at the end, but you have to invest 2 in the beginning, which is an endergonic reaction because you need to put energy in. It can't happen randomly. How much does link reaction give you? 0. Krebs cycle. It gives you 2 ATP, and the ETC, 32 ATP. So in physical form, glucose is formed into what during glycolysis? What is the final reaction? Pyruvate, and then acetyl-CoAs, and then acetyl-CoAs then turn into carbon dioxide. And at the very end, you could also turn that into NADH and FADH. They carry the electrons to make ATP and can also form water. So what is the total amount of ATP formed here? NAD plus is a coenzyme. What is a coenzyme again? is a cofactor. Coenzymes are cofactors, they're just organic. What is the difference between fermentation and aerobic respiration? Aerobic respiration uses oxygen in its reactions, and fermentation is the partial degradation of sugars without the use of oxygen. So glycolysis is fermentation because it's the degradation of sugars. Degradation of sugars without the use of oxygen. Aerobic use of oxygen. The breakdown of glucose in cellular respiration has a positive amount of energy, yields a positive amount of energy at the end, meaning this reaction has a positive or negative free energy yield. A negative energy yield, an exergonic reaction in total, because it can happen spontaneously. It yields carbon dioxide, water, and energy. Don't even listen to this. Just remember that the breakdown of glucose has a negative free energy yield and an exergonic reaction, and it yields carbon dioxide, water, and energy. You get energy from ETC, water, carbon dioxide. To enter the citric acid cycle, pyruvate must enter the mitochondria by active transport. Three things are necessary to convert pyruvate to acetyl-CoA. Complete the missing parts of the following figure, and then explain the 3 steps of the conversion process. So I see here 2 carbons. This being the pyruvate, CO2 is taken out, so 1 CO2 is taken out, and a coenzyme goes in here because there's 1 carbon left, and through this NAD plus is reduced to NADH because reduced is gaining. So there's just a visual for you. Pyruvate carboxyl group is removed and given off as a CO2 molecule. The remaining 2 carbons. Oh, pyruvate is 3 carbons, my bad. 1 carbon is given off as CO2. The remaining 2 carbons are oxidized, which means losing, forming acetate, the extracted electrons. Because they're oxidized, are given to NAD+, storing energy in the form of NADH to later give to the ETC. A coenzyme derived from vitamin is attached via its sulfur atom from the acetate. This high energy potential will now feed its acetyl group into the citric acid cycle. The citric acid cycle is another name for the Krebs cycle. Oxidative phosphorylation involves 2 components, the electron transport chain and ATP synthesis. Notice that each member of the electron transport chain is lower in free energy than the preceding member of the chain, but higher in electronegatividity. The molecule at 0 free energy on this chart, which is oxygen, is the lowest of all the molecules in free energy and the highest in electronegatividity. Remember, oxygen is high in electronegatividity because you remember that from water, because it draws in the hydrogen for the polar covalent bonds, the uneven sharing. Why is oxygen considered the ultimate electron acceptor? Because it is extremely electronegative. It wants to take it on, those 2 electrons. Oxygen stabilizes the electrons by combining with 2 hydrogen ions to form 1 compound. Going back to what we reviewed in the beginning, and in its 1 and 2 video, H2O, it forms water. Everything comes back to water. Alright, we did that. If you guys aren't even asleep, photosynthesis, which yields, what does photosynthesis make? Glucose and O2, which helps drive the Calvin cycle. Glycolysis, I believe. Never mind, I'm not gonna. Alright, let's hear here. What are the stacks? Where does photosynthesis happen? A chloroplast. In the chloroplast, what are these little discs? Not the stacks of discs, but the individual discs themselves, where the light reactions happen. They're thylakoids. Photosynthesis happens in the thylakoid. What happens in the chloroplast, but thylakoids is where the light reactions occurs. This stack of thylakoids is called what? Granum. It's called the granum. And what in light reactions is used? Like how is it? What does it do? It takes in water and light. An ADP and NADP. And using the light energy, it splits the water, which is used in photosynthesis and ETC, to create a proton gradient to produce ATP and reduce NADP to NADPH. So the light energy splits the water, which creates a proton gradient to produce ATP and reduce NADP. And then O2, because when water splits, you get the oxygen and hydrogens. That's when it splits. Where does this happen? Where do light reactions happen? The thylakoid membrane. Where does the Calvin cycle happen? The stroma. It happens in the stroma, which is it's like the cytoplasm of a chloroplast. What does photosynthesis do, like overall? Converts solar energy to chemical energy, which is glucose, and then molecules produced of waste happen to be O2 as well. What is photolysis? The splitting of water using light energy. What is photorespiration? Which happens in the Calvin cycle. When the enzyme Rubisco, which is a carbon fixation kind of enzyme, binds to the RUBP rather than the CO2 in the Calvin cycle. This occurs in dry conditions when the plant closes its stomata. What is a stomata? They're little holes for the oxygen and CO2 to go in and out of the leaves. The slow water loss via transpiration. And allows water to be transpirized. Stomata. This traps O2 gas inside because oxygen also uses it. The plant, and when O2 concentration is higher than CO2, Rubisco favors O2. So photorespiration inhibits the Calvin cycle, and a problem that needs to be addressed in dry climates. So remember, photorespiration is a problem if the stomata closes in dry environments. It keeps the O2 inside. Rubisco favors O2 instead of CO2. What is chemiosmosis again? The flow of hydrogen ions through their concentration gradient through ATP synthase. What is the difference between cyclic and non-cyclic photophosphorylation? This is the use of light energy, photosystems, and ETC to create an H+ gradient in order to produce ATP, but PL replaces electrons lost in P2 compared to non-cyclic, where water replaces the P2. Photosystem 1 replaces electrons lost in photosystem 2, creating a cycle. Non-cyclic phosphorylation happens when water replaces the electrons lost, and it just keeps on going. What photosynthesis looks like. Photophosphorylation. The stroma, the thylakoid space, is happening in the thylakoid membrane. Light reactions, photosystem 2, photosystem 1, water going in, oxygen coming out. Where is the electron transfer taking this? It's going to be from here to here. The ETC right here, this is the ATP synthase kind of thing where chemiosmosis occurs, generating that ATP, which ATP synthase right here, spinning this little thing, which I believe is like a motor. Chemiosmosis refers to the energy coupling mechanism that uses energy stored in the hydrogen across the membrane to drive cellular work like ATP synthase. Proton motive force refers to the potential energy stored in the proton-electron gradient generated by the pumping of H+ ions across an optical membrane to whatever you got from that, just take it. Thylakoid space, stroma, started with photosystem 2, photosystem 1 can replace the electrons lost here, giving it, they can recycle that to create a cyclic photophosphorylation. Where is photolysis? Photolysis happening in the diagram, which is the splitting of water using light energy. Photosystem 2 is where it's getting that extra electron, the hydrogen from the water, releasing O2, and then sending it through the ETC to photosystem 1, where after the ETC it takes that H+ ion, puts it through ATP synthase, making ATP through phosphorylation, and then taking an extra electron after photosystem 1, turning NADP, reducing it to NADPH, and then that can go back and replace the electron in photosystem 2, or water can just replace it, creating non-cyclic and cyclic. Now for the Calvin cycle. molecule is regenerated in this regeneration phase that is then used in the carbon fixation phase, making this a cycle. Carbon fixation phase, reduction phase, regeneration phase. It would be 5 carbons in the RUPP because 3 ATP is made here. Often the enzyme comes into play, Rubisco, carbon fixation. After it gets carbon fixated, it takes Rubisco. There's reduction of ATP to ADP, NADPH, and NADP. Pretty much, this is the ETC of photosynthesis. And it takes NADPH, NAD+. It's going to bring that back around and give it back to photosystem 1 and 2 where they need to use it, and regeneration occurs after glucose is made. And then the G3Ps are recycled. And you start back with 3 RUPPs. 3 RUPPs. Where are they? 3 RUPPs. Otherwise it's like weird. Okay, that's all we're gonna do. Actually, here's a better sketch. Photosystem 2, photosystem 1, photosystem 2. Electron sun energy breaks the water bonds. Water goes in. O2 comes out. Electrons go out, go through. It powers it. There's some electron acceptors in there. It eventually makes H plus gradient. It's taking those electrons, putting it down there. There's another electron goes here. And then NADP is reduced to NADPH right there. Hydrogen gradient creates ATP, and then ATP and NADPH goes to the Calvin cycle to make glucose. I didn't even make it to unit 5 yet. And this video is an hour long. Thank you, Emily, so much for letting me use your phone. But I hate this. I'm not gonna learn it anymore. Okay, I'm gonna really try my best to do units 5, 6, and 7 tomorrow. Because I'm not feeling 5 right now. I don't think I could teach it very well. I couldn't even remember what I was teaching with photosynthesis recently, so I'm gonna try and upload this video right now. Hopefully I can be in bed by 2. And then I'm gonna try and be in bed really early tomorrow because the AP test is then. So yeah, but thank you for watching. Good luck on your AP tests if there's no upload tomorrow, because I have a feeling that might happen. But yeah, thanks for watching.